get time to realize it's right before my eyes, and I can take it if it's what I want to do. I am leaving, and this is starting to feel like it's right before my eyes, and I can taste it. It's my sweet beginning. I can tell just what you want. You don't want to be alone. You don't want to be alone. I can't say it's what you know, but you known it the whole time. Yeah, you known it the whole time. Maybe next year I'll have no time to think about the questions to address. Am I the one to try to stop the fire? I wouldn't test you, I'm not the best you could have attained. Why try anything? I will get there. Just remember, I know that I can tell just what you want. You don't want to be alone. You don't want to be alone. And I can't say it's what you know, but you known it the whole time. Yeah, you known it the whole time. I can tell just what you want. You don't want to be alone. You don't want to be alone. And I can't say it's what you know, but you known it the whole time. Yeah, you known it the whole time.
Okay, good morning, everybody. So today is April 22nd, Wednesday, and we're going to be going through Hardy-Weinberg equilibrium. Let me pull up my script. Okay, so remember that all populations are kind of evolving, and when we measure this, we're measuring it on the population level, not necessarily on the global level. And at its core, evolution is at its core, it's a change in the genetic makeup. All right. Even though, yes, it does act on the phenotype is influenced by the genotype. All right. So let's say there is let's say there's a trait that is environmentally influenced. Let's say I don't know. The animals that we had talked about previously that have the ability to change the coloring of their fur, but they don't have direct control over it, and it changes based on time of year. If that is not controlled genetically, then that means that selection against it isn't going to change anything about that population. All right, so remember that we're looking at these genetic changes because we need to be able to pass it on from the parents to the F1 to the F2 generations. All right, so this has to be something that that we're passing down. So going through, we're going to start looking at what is called Hardy-Weinberg. All right, we're going to start looking at Hardy-Weinberg. And so Hardy and Weinberg, they were two scientists who kind of made this conception that you can measure the proportion of alleles in a population and kind of measure that frequency and measure it over certain generations. And so Hardy-Weinberg equilibrium is when a population is not evolving. All right, so it's when a population is not evolving, but it still looks at allele frequency. So just because a population is not evolving doesn't mean that we're going to get 25%, 50%, and 25%. That's not what that means. All right. So it is not this. What it means is that whatever these numbers are will stay consistent over time. All right. So they stay consistent. All right. This is kind of the key to it, because if these numbers start to change, then that's when you're out of Hardy-Weinberg equilibrium. So what is an example of this? So first we have to look at the Hardy-Weinberg equation, which is p squared plus 2pq plus q squared equals 1. All right. Why does it equal 1? Well, because remember, 1 is 100%. So we're looking at this as a proportion rather than looking at it as a percentage. So if I say 0.3, you know that 0.3 is 30%. All right, so 0.3 comes out to 30% in this situation, and 1 comes out to 100%. So the way to check your math on this is to make sure that everything adds up to 1. So let's use a hypothetical population. All right. So I'll open up a new thing for this. So we'll use a hypothetical population. Let's say in this population you have so as a reminder, the equation that we're using, p squared plus 2pq plus q squared equals 1. And just to kind of clarify so that it's p is your frequency of your dominant allele, and q is your frequency of your recessive allele. All right. So let's go to a population of beetles, right? We have a very large population of beetles. Some are dark gray, which we'll assume is the dominant allele, and then light gray is the recessive allele. So if I say that the frequency that this occurs is 0.3, instantly, what is the recessive allele if that's the only information I give you? Well, it has to be 0.7. Like, you have to make up for that value, because both of these together have to equal 1. That's a really bad way of putting it. Let's do it. Like, they have to equal 1.0. All right. So they have to come out to equal 1. So if you only know just one half of the information, then you should be able to figure out the second half. So doing a Hardy-Weinberg equilibrium, what you can do is, because you know this, you know that p squared is 0.3 squared, and that gives you 0.09. All right, 0.09. you know that 2 PQ, if you fill this in, it is 2 times 0.3 times 0.7. All right. And then I'm going to show how this is done with a calculator. Where's All right, so if we have 2, we multiply that by 0.3, and then we multiply that by 0.7, we get, it should be 0.42. Oh, it was trying to convert it for me. So you could see how we ended up with 0.42. All right, so red 0.42. And then if we do Q squared, which is 0.7 squared, so using our calculator, 0.7 times 0.7 gives us 0.49. All right, 0.49. So what is this actually saying? Like, why are we doing this other than just to have some math to do? So what is the whole purpose of plugging all of this in? What we can assume out of this is, in a population, if you know that P squared, or P is your dominant allele, then P squared has to be your homozygous dominant individuals, Q squared has to be your homozygous recessive individuals, and 2PQ are your heterozygous individuals, which means that in your population, you have 9% that is going to be the dark black color, or the dark gray color, which is homozygous dominant. You know that for your heterozygous individuals, you're going to have 42% that is heterozygous, and that you're going to have 49% that is homozygous recessive. All right. And that tells you a lot about this population. You could start to make assumptions or predictions off of, you know, how is distribution of alleles going to occur in the future, because you now know what's out there. You know that a majority of the time you're probably going to have one of these individuals, a homozygous recessive, mating with one of these other individuals. How do you know that? Well, because almost 50% of the population is going to be made up of this individual. So it would be like if I said, you have to reach into a container that contains 100, let's say there's 100 pieces of paper in it. Each of them has one of these individuals on it. All right. You reach into so in this box, 109 of them are homozygous dominant, 42 are heterozygous, 49 are homozygous recessive. You go into this box and I tell you, you have to pull out 2 individuals. All right. You pull them out, those 2 individuals will then mate. Most likely, you're going to pull out individuals in this. All right. It's going to be one of those 2 individuals. This will be a little bit more rare, but for the most part, it's going to be one of these 2 individuals right here. All right. And why is that significant? Well, because what it says is that 91% of the time, you're going to pass down one of these alleles. And the dominant allele in there tends to be your homozygous recessive. So going back to these numbers, that means that out of all the mating events that happen, 0.7 will be this, and then 0.3 will end up being passed down as your dominant allele. All right. So we can use this to make assumptions about what the next generations are going to look at. That also tells us a little bit about the past of these organisms. there was a selection event previously. Well, how do we know that there was a selection event previously? Well, if this is dark colored, all right, if this is dark colored and this is light colored, or dark and light gray, we know because this percentage is low that there had to be a selection event on that causing most individuals to move toward the light individuals. But that doesn't explain how this is 42%. Well, over time, given random mating, these numbers would have started to shift a little bit more because you're just giving one of these alleles back to whatever's left in this population. So you could make some assumptions about what happened to a population previously based off of your Hardy-Weinberg equilibrium. All right. So because of this, we could say that they are in Hardy-Weinberg equilibrium. So let's make a prediction about what would happen if they reproduce the next generation. What will be the alleles in the genotype? A lot of this we can predict. So first, we have to assume that none of these genotypes are better than the others. So going back to our example with the hawk and the mice, we have to assume that that is not happening. Second, we have to assume that they mate randomly. All right. If there's any sort of mate selection, so for mate selection, look at cardinals. All right. So with cardinals, bright red is an advantage. All right, bright red is an advantage. Is this in Hardy-Weinberg equilibrium? No, because there is a selection process that's going on. So this is said to not be in Hardy-Weinberg equilibrium because mating is not random. And then what we could do after that is we can modify a Punnett square. All right. So this is kind of adding a little bit onto it, but if you have random mating, and we're going to use heterozygous individuals in this instance. If we make it proportional, you end up with something that looks like this. Why do we end up with this? Well, this is going to represent your 0.49. These are going to represent your 0.21. Yes, even though it looks like these are smaller boxes, it still does come out to 42%. And then this is going to be your 0.9. So let's color code this. All right, let's color code it. So let's say that you have blue here. Say these boxes are green. And then here's your red. All right. So you could see that the size of your box represents the distribution of your alleles. And these two, the heterozygous individuals, they will always come out exactly the same. All right. They'll always come out to exactly the same size because you're going to be adding them together. Why are they 21%? Well, you have 42 divided by your two boxes, and that gives you your 21%. All right. So this is all based off of random mating. All right. So is this completely realistic? Not really. You could see it on a very short scale, but long term, this does not tend to be super realistic. Why does it not tend to be realistic? Well, because Hardy-Weinberg has some assumptions. All right, so there are some assumptions, and there's basically 5 of them. So one is no mutations. So there are no mutations at all. Mutations are random changes that happen in DNA. Well, random being used loosely. It's random where they take place at, kind of. Like we talked about, if you are exposed to UV light, like people that go outside and they're exposed to a lot of UV light, you can end up with thymine dimers, which is where these will bond together. And when they are read, they actually read as one letter rather than two separate ones. All right, so that's why they always say wear sunscreen outside, because it can lead to thymine dimers. So one is no mutations. Two is one that we had already talked about, that there is random mating. All right, there's random mating. You can get random mating in a lot of populations. Insects, for instance. Insects tend to have quite a bit of random mating. There are some that don't. There are some that have selection. But look at very novel traits that are selected for. We talked about the cardinal earlier. Does a cardinal being red or bright red or having the best coloring say anything at all about its fitness? No, it doesn't. All it says is that I have bright red coloring and bright red feathers, and therefore that's a trait that's selected for. At the Fort Wayne Children's Zoo, one of the things, whenever it opens back up, you can go there and there is an albino peacock. It is completely white, and it's a male, and males have the long feathers. And if you've ever seen a peacock do its mating dance, it fans its feathers upward, and then it kind of bends them forward, and then it shakes them in order to kind of make this rustling noise. And that's it doing its dance in order to try to attract a female. If you go there, you will see the male peacock that's albino pull its feathers up and start doing its mating dance. But it will not mate, because it doesn't have that bright coloring, and therefore females aren't attracted to it because it doesn't have that bright coloring. To us, it actually looks really cool. Like if you've never seen it, it does look really, really cool. The other thing is that there is no gene flow. And gene flow is gametes or alleles entering or exiting a population. So think about it as you have a group of flowers out in front of your house. Your neighbor across the street has the same type of flowers. You selected yours for a specific reason. They selected theirs for a specific reason. If the wind blows and blows pollen into your direction, or if an insect gets onto their flowers and brings that pollen over to yours, that is gene flow. All right, so think about it as a river flowing. As a river flows, it's going to bring stuff from one side, and it's going to make its way all the way down to the other side. So think about genes flowing down a river. This is where it's supposed to be, and this is where it ends up at. All right. The fourth is that you have a large population. Large population size. Why is large population size a big deal? Well, because if you have a smaller population size, it's going to start to reduce this random mating. The other thing is any mutations are going to completely shift the dynamics of that population. So the way that we've always kind of talked about this is, if you have Hardy-Weinberg equilibrium, you have to assume that you have a near infinite population. And we know there's no such thing as near infinite, but you have to think about, like, if you've ever seen a cluster of mosquitoes or insects as they kind of cluster around and fly together and things like that. That, even though that is a finite amount, you could think of how many individuals are in there, and if mating is random 100% through all of that, then that would be Hardy-Weinberg. And then the last assumption is no natural selection. All right. No natural selection. So thinking about the example with the mice where you had, actually pull that example back up. Where's my mice example at? With this, in this example, so you have dark colored mice, light colored mice. The light colored mice are selected against. The world's largest truck decided to drive by my house with the most annoying exhaust ever. So you have kind of these random, or this spread of individuals. You can see a majority of them tend to be the tan colored, and then you have the black colored mice as in the minority here. But then that shifts into the end because the tan colored are selected against, and now the black mice have a majority and the tan mice have a minority in this population. So the alleles have shifted in this case. This is selection. This cannot happen in Hardy-Weinberg. You cannot have that situation. There has to be zero selection. So what happens if you fail one of these? Well, if you fail one of these, that means that the population is not in Hardy-Weinberg equilibrium. All right. So that means that there's some sort of evolution. As you move on to the next generation, your proportions of that population are going to change. And it's one of those things like if we know that very few populations meet Hardy-Weinberg equilibrium, why do we do it? Well, we do it because that's the way it's always been done. And that I know that is the worst way to phrase anything with Hardy-Weinberg. Talking to a friend of mine that she is a evolutionary ecologist, one of the things that she said is that when she did her research, she knows 100% that it is not in Hardy-Weinberg equilibrium because there was a selection event, but you still have to do Hardy-Weinberg in order to show that it failed that. Why? Because everybody does it. It's kind of like myself as a botanist. If I do any sort of research that involves plants, I have to make an annotated species list at the very end if my research involved more than one plant. Like if I'm looking at a community level, I have to include every single thing that I came across. Why? Because everyone does it. And so Hardy-Weinberg is one of those things that everybody does it. So we just keep doing it. And it's a really bad reason to continue doing it. But remember that not all genes satisfy Hardy-Weinberg. Some do, some don't. So you can think of it in two ways. So looking at one gene or for all the genomes. If we look at just one gene, we could check whether the above criteria is true for that one gene. So ask yourself if there were a mutation for that gene or if the organism randomly mutated with regards to their genotype for that gene. So using, let's say, eye color as an example, would that have Hardy-Weinberg equilibrium? Well, yes, there's a mutation in there. But what if it was something like hair color, where there's no mutation, there's a plenty large population size for it, we're not selecting for or against it, mating's random. Yes, we have gene flow, but we have gene flow for, we're a global species, so we could just say that there's no gene flow. And therefore, we could say for something like hair color that it is in Hardy-Weinberg equilibrium. But remember, if we look at all genes in the genome, the conditions have been met for, or are these conditions met for every single gene? And it is possible for them to be met with, or for these conditions to be met for some of them under certain circumstances. But it's unlikely that you're going to see this with all genomes all over the world. All right. Some populations might have Hardy-Weinberg for some genes and others might not. Right. So for example, let's go back to the flower example of, my flowers versus my neighbor's flowers. I've selected mine for a certain instance. He selected his for a certain instance, but due to the proximity, there's going to be very little, or there's going to be a lot of gene flow between those. Also, the population size isn't all that big. But then take an area like Eagle Marsh or Arrowhead Prairie. very large place, large population size. There's no real selection going on. There's not much gene flow since it's surrounded by agricultural land. And mating is completely random. So in that situation, yes, you would have Hardy-Weinberg equilibrium. All right, so you would have Hardy-Weinberg equilibrium. So what this does is this leads into what are mechanisms of evolution. All right, this leads into the mechanisms of evolution. So what do I mean by that? What causes a species to actually change? Well, we talked about, I mean, let's go through our Hardy-Weinberg list. No mutations. So do you want to guess what the first mechanism is? And if you guessed it is mutations, you are correct. So all a mutation is where you change something in a population. All right. So I'm going to pull up an example really quick. Here's an example of a mutation. Notice you have your frequency of all of your alleles come out. They're all 100% heterozygous recessive. Instantly during this process, you have one change. Now there is a dominant allele in this. You have changed your Hardy-Weinberg equilibrium, or you've changed your numbers, because now you have one individual that contains one dominant allele, and therefore you have this new genotype, and you've changed your frequency. This is what a mutation is. It's just a change that happens. Now this says random mutation. Mutations can sometimes be directed based off of the mechanism of action of the mutagen. And one of the things that when we were in class that I really like to talk about is mutagens. Not so much going to discuss it now, but we did hit on mutagens quite a bit. All right. So mutations is one. The second one is non-random mating. All right. And sometimes we call this mate selection. So thinking about there's a type of bird. The documentary is online, but there's a type of bird. Here comes my great bird drawings again. All right, that's a bird. And what it does is it builds this nest. So here's a tree branch. It builds this like hanging nest. All right, it builds this like hanging nest thing that the bird kind of perches in. All right. And then what happens is a female bird flies in and lands in the nest. And the thing is that usually the first year they don't have much experience building this. And in this documentary, the female bird flies in, lands, and then this thing breaks and flies to the ground, and then she flies out. But over time, he gets better at building nests due to experience. All right, so he builds better and better nests due to experience. So then the next year, so this is year 2. The second year, the bird flies in and lands, and it may or may not hold up. But let's say it holds up. Well, then that bird gets an opportunity to mate. So you've instantly introduced a mechanism into mating based off of selection. So here's an example. So assortative mating is where they prefer similar genotypes and phenotypes. So these dark colored beetles and light colored beetles will mate together, while disassortative basically means that it's random mating. In this situation, there's going to be a selection pressure for these specific alleles within these populations, while in the dissociative, there is not. There's other types of things. Bullfrogs being able to call loudly says nothing about their underlying genetics or anything like that. So those would be mate selection. All right. The next one is gene flow. So in the example of gene flow, you have, let's say we go back, mating is random. You have these darker colored insects and these lighter colored insects. One of the dark ones decides to make its way over this geological feature into this population. And once it makes its way into that population, excuse me, there is mating that occurs, and therefore these genes are now introduced into that population, causing a change in your proportions. And that can be one of two things. All right, so you can look at this as the movement of individuals. This one individual moved over here, but think about this: if these were flowers and this was pollen moving. So it doesn't necessarily have to be the organism. It just has to be movement of those alleles. All right. It has to be movement of those alleles. And gene flow is extremely strong in evolutionary pressures, because what you are doing is, think about it as cooking. You are adding new ingredients into the pan that did not exist there before. Therefore, you change the entire flavor of the meal. In this situation, you are introducing genes that were not there before, and you are going to change that population. All right. So that is an extremely strong pressure when it comes to this. The next one, what did we have? We had okay, so non-random mating. We had gene flow. I didn't write it here, but gene flow for those of you that are taking notes. So this can be an individual or gametes, like in the case of pollen. All right, individual or gametes. The next one is can be what's called a I guess non-infinite population size. Does anyone ever call it this? No, we call it genetic drift. All right, we call it genetic drift. But what genetic drift is it's basically where your allele frequency changes due to chance events. So it can be sampling errors. It also could be if you have small populations and it's, you know, out of that small populations, a lot of times you're going to have shifts one way or the other. And what that means is a drift in, if your allele frequency is centered, now instantly your allele frequency is off-centered in either one way or the other. So there's a drift to one of these two sides. All right. Then I'm gonna have to move over here for the last one. The last one is, back to my red. The last one is natural selection. Making this fit. I know it'd be easier to draw it below it, but we're doing this. Natural selection. There we go. So natural selection is going back to that the bird and the mice examples, where you have these individuals that are up against this this dark colored rock, the darker colored mice are going to be an advantage, and therefore the light colored mice are going to be selected against due to predation. There can be other things with this. It could be disease that selectively targets certain groups of individuals. It all depends on what your environment is. One of the kind of the classic examples is the peppered moths. I know people are tired of hearing about the peppered moths, but I know that can be argued that that wasn't natural selection. It was artificial selection, since the Industrial Revolution changed the coloring of the environment, therefore differentially selecting against those individuals. A lot of people argue that it's natural selection, that yes, even though we did change it, those genes exist naturally, and the birds going after the individuals was a natural process. So there is debate going on to one side or the other. But that is pretty much everything that I have for today for Hardy-Weinberg. I know it seems like it's a very abstract concept, and the reason it's a very abstract concept is because it kind of is. Like, we're referring to different traits, different genes, different things that are happening within an environment, and you're trying to satisfy this kind of odd list of things that we know not all populations and not all genes are going to interact with. And this kind of it goes back to a lot of the concepts where we talk about in science, where what is or how should something work. Like, we talk a lot about the law of conservation of matter and the law of conservation of energy, where it's nothing is created or destroyed, and energy is not created or destroyed. It can only change. And same thing with matter. It cannot be created or destroyed. It can only change. Yet we know that, like, as that works out, but in a closed system. So is there such thing as a closed system? I mean, theoretically, the universe is a closed system, but looking at our planet, no, it's not. We lose matter every single time we throw something up into space, and we don't get it back. And there's these, I know it's like a small fraction of the overall matter that there is on the planet, but looking at Hardy-Weinberg, is this something that could happen? And in theory, yes. In theory, a lot of this stuff should work out, but in practice, this a lot of times isn't what we see. And kind of evolution is this big process of trying just to disprove everything in evolutionary biology, or is trying to I'm sorry, let me rephrase it. Evolutionary biology is trying to disprove things for Hardy-Weinberg, or to try to find the exceptions. So, for example, when the friend of mine, when she was doing her research, one of the things that she was trying to look at was, how did gene flow and genetic drift happen over this known event, and how did that affect the genetics of this population? Because there was a known event. Now, it might have been random selection, but they're not entirely sure. So they wanted to see, how did their genetics change before and after? And granted, we don't have before genetic information, but we do have such a large sample with after genetic information that we can kind of take guesses as to what it was beforehand. And so this is kind of the whole purpose of Hardy-Weinberg. Let me turn my light on and switch over to my camera so I can do desktop. So I know that this seemed like it was a really confusing lesson. So for next time, what we're going to do is we're going to do an example over genetic drift, and kind of talk about founder effects and bottlenecks, which is one of my favorite things in evolutionary biology. Then with that, the next one after that is going to be looking at natural selections in populations, how looking at directional disruptive selection, stabilizing, things like that. And then we're going to move on into species and speciation. What makes a a species different. So looking at the African fish eagle versus a bald eagle, they look very similar, yet they're different species. But looking at a Great Dane versus a Chihuahua, there's the same species, even though they look vastly different. Looking at when we make hybrid, or when hybridization happens, and then the different barriers of what can and can't stop things from creating a new species. Then we're going to go through phylogenetic trees, and then I'm going to post a test onto PowerSchool. If you do that test and test corrections are afforded, I will use that to replace an old test grade. All right, there will be a limited amount of time where you can take it. Probably what it'll be is, instead of me sitting here doing lectures, I'll open the test up, and then you'll be able to take that test on that. Then after that, we're going to move back into not necessarily ecology, but it's going to be more of like the human global impact, which is a little bit of a lead into kind of environmental science. But it's there are certain things that we're going to hit on. On PowerSchool, there is a quiz, or I'm sorry, it's an assignment. I want to say it's 5 questions, something like that. Let me see if it posted. I was having problems trying to post it. For some reason, it is not posted, so I still have that on my computer, so I will repost that. That is under the assignments tab. I'll show you guys how to actually do this, since there's a lot of confusion with this. So let me switch over to my window. Okay. Looking at this is what you guys see when you load up. So here's the schedule. For some reason, you guys spend a lot of time looking at my picture. Office hours, when that's going to be, a link to the YouTube channel, and then the syllabus, and then the course description and biography, which has been there. If you go over here to the side to assignments, we haven't gone through all of these. So if you're like, do I have to do every single one of them starting at the beginning? No. But for instance, we did go through mitosis. So if you are missing mitosis, do this. The other thing is these assignments over here. These are all the assignments we went through, and then all of the new stuff that I've been posting. So if you want to do these assignments in order to improve your grade, click on it, download it, put it into Office 365, work on it, share the link with me when you're done. Use your school email, please. I am getting so many emails from people's outside email. All right. And it's the thing is that I understand that might have been an email address that you made a long time ago. And like I said before, I have emails that I used a long time ago, but now you need to, start acting like you're moving into your adult years. Use your school email. I don't want to get something from like Christian is the man 42069XX at outlook.com. Just send it from your regular school email address. Don't send something stupid and ridiculous. A lot of times that stuff will end up in my junk box, and then I don't ever see it. So make sure that you're turning everything in based off of your school email account. The other thing too is make sure that you're formatting emails properly. Don't send me an email that sounds like you're talking to one of your friends. You have time to format it and things like that. Put in a subject, even if it's just question or help or something like that, and then type out the body of the message. Hey, I have a quick question about the assignments. Which one of these am I supposed to be doing? And then I can answer that. But here's where all of your assignments are. For some reason, the new one isn't loading up, so I will get that to load up really quick. Switch back over to this. I'm going to actually load it right now. But that is kind of the plan for the rest of the school year. If you guys have questions about any of that stuff, or if you're having problems, send me an email. I do office hours directly after this, and for some reason, no one ever shows up to those office hours. Okay, it says it's still being copied and it'll be available soon. So hopefully that'll show up soon. show up to office hours to ask questions. If you can't make it, send me an email. I'm available. If you want to talk one-on-one rather than through email, send me an email saying, hey, can we schedule a time where we can meet and talk about certain things, and we can sit down and do a private Google Meet, Skype, Zoom, whatever you want to do. I'm open for whatever. I'm providing you guys with the tools to do well in this course. You guys just have to utilize those tools in order to improve your grade. So make sure that just because we're out of school doesn't mean that we're out of school. Like, we're still doing this. We're still, like, everything is still going on exactly the same as it was before. So don't just assume that because, you know, your buddy's not doing anything, that means that we're out completely. All right. So that's going to be the end for today. Again, if you guys have any questions, email me, and I will be in Google Meets. I'll post the link immediately after this, but I'll be in Google Meets until 11.